Welcome to our exploration of emerging technologies in systems biology. Systems biology is an interdisciplinary field that focuses on complex interactions within biological systems, aiming to understand how these interactions give rise to the function and behavior of living organisms. In recent years, technological advancements have revolutionized this field, enabling researchers to analyze vast amounts of biological data and uncover new insights. Today, we'll journey through the latest innovations shaping the future of systems biology, from high-throughput data generation to artificial intelligence-driven analysis. Let's begin by understanding the foundation of systems biology and why it's so crucial in modern science. One of the biggest drivers of systems biology has been the rise of omics technologies. Genomics, proteomics, transcriptomics, and metabolomics allow scientists to study genes, proteins, RNA, and metabolites on a global scale. These high-throughput technologies generate massive datasets, providing a comprehensive view of biological processes. The integration of these omics layers is essential for understanding the complexity of life. As we move forward, improvements in sequencing speed, accuracy, and cost continue to expand the possibilities for systems-level investigations. The explosion of omics data, systems biology has become a big data science. Managing, storing, and analyzing these enormous datasets requires advanced computational tools and infrastructure. Cloud computing and distributed databases have become essential for handling the scale and complexity of biological information. These technologies enable researchers to collaborate globally, share data seamlessly, and accelerate discovery. The ability to process and interpret big data is now at the heart of systems biology research. Artificial intelligence, AI, and machine learning are transforming systems biology. These technologies can identify patterns in complex datasets that would be impossible for humans to detect. AI algorithms are used for tasks like predicting protein structures, modeling metabolic networks, and identifying disease biomarkers. As AI continues to evolve, its integration with systems biology promises to unlock new levels of understanding and innovation, driving the field toward more predictive and personalized approaches. Single-cell analysis is a groundbreaking advancement in systems biology. Unlike traditional methods that average signals across thousands of cells, single-cell technologies reveal the unique characteristics of individual cells. This approach uncovers cellular heterogeneity, helping scientists understand development, disease progression, and treatment responses at an unprecedented resolution. Techniques like single-cell RNA sequencing are now widely used to map cell types and states in complex tissues. CRISPR technology has revolutionized genome editing, allowing precise modifications to DNA. In systems biology, CRISPR is used to perturb genes systematically and observe the resulting effects on cellular networks. This enables researchers to map gene functions and interactions at scale. Genome editing tools are also being combined with omics and AI to accelerate functional genomics studies and therapeutic development. Mathematical models are essential tools in systems biology. They help researchers simulate biological processes, predict system behavior, and test hypotheses in silico before conducting experiments. These models range from simple equations describing gene regulation to complex simulations of entire cellular networks. Advances in computational power and algorithms are making it possible to build more accurate and detailed models, bridging the gap between theory and experiment. Work biology is a core concept in systems biology, focusing on the interactions between genes, proteins, and metabolites. By representing these relationships as networks, scientists can identify key regulators, modules, and pathways that control biological functions. Network analysis reveals how perturbations in one part of the system can ripple through and affect the whole organism. This approach is crucial for understanding diseases and designing targeted intervention. Synthetic biology combines engineering principles with biology to design and construct new biological parts, devices, and systems. In systems biology, synthetic biology tools are used to test models, build synthetic gene circuits, and create organisms with novel functions. This integration accelerates our ability to understand and manipulate biological systems, opening new possibilities in medicine, agriculture, and environmental science. Biological systems operate across multiple scales, from molecules to cells to organs and organisms. Multi-scale modeling aims to integrate data and models at different levels, providing a holistic view of biological processes. This approach helps researchers connect molecular mechanisms to physiological outcomes, improving our understanding of health and disease. Emerging computational frameworks are making multi-scale modeling more accessible and accurate. Spatial omics technologies are transforming our ability to study the organization of molecules within tissues. By mapping the spatial distribution of RNA, proteins, and metabolites, scientists can uncover how cellular context influences function. These technologies are particularly valuable for understanding complex tissues like the brain and tumors, where spatial relationships are critical. The integration of spatial data with other omics layers is a major frontier in systems biology. High-content imaging combines automated microscopy with advanced image analysis to extract quantitative data from biological samples. This technology enables large-scale studies of cell morphology, protein localization, and dynamic processes. High-content imaging is often integrated with omics and computational analysis, providing a rich multidimensional view of biological systems. It's a powerful tool for drug discovery and functional genomics. Wearable biosensors are bringing systems biology into everyday life. These devices continuously monitor physiological parameters like heart rate, glucose, and metabolites, generating real-time data streams. Wearable biosensors are enabling personalized health monitoring and early disease detection. As sensor technology improves, the integration of wearable data with other biological information will provide deeper insights into human health and disease. Lab-on-a-chip technology miniaturizes laboratory processes onto microfluidic chips. These devices can perform complex analyses with tiny sample volumes, making high-throughput screening faster and more efficient. Lab-on-a-chip systems are used for genomics, proteomics, and diagnostics, enabling rapid and cost-effective experiments. Their portability and automation are transforming both research and clinical applications in systems biology. Cloud-based platforms are revolutionizing how scientists collaborate and share data in systems biology. These platforms provide secure, scalable storage and computational resources, allowing teams to work together across the globe. Cloud-based tools also support reproducible research by standardizing workflows and data management. As collaboration becomes more digital, cloud technology will be central to the future of systems biology. Data visualization is key to making sense of complex biological data. Advanced visualization tools help researchers explore multidimensional datasets, identify patterns, and communicate findings effectively. Interactive dashboards, 3D models, and network graphs are just a few examples. As datasets grow larger and more complex, intuitive visualization will remain essential for discovery and decision-making in systems biology. Open science initiatives are promoting transparency and collaboration in systems biology. By making data, methods, and results publicly available, researchers can build on each other's work and accelerate progress. Open data repositories and preprint servers are becoming standard practice, fostering a culture of sharing and reproducibility. This openness is driving innovation and ensuring that discoveries benefit the broader scientific community. Personalized medicine is a major application of systems biology. By integrating genetic, molecular, and clinical data, scientists can tailor treatments to individual patients. This approach improves outcomes and reduces side effects. Emerging technologies like AI, single-cell analysis, and wearable sensors are making personalized medicine more precise and accessible, transforming healthcare for the better. Digital twins are virtual models of biological systems that mirror real-world processes. In systems biology, digital twins can simulate organs, tissues, or even entire organisms, allowing researchers to test interventions and predict outcomes. These models integrate real-time data from sensors and experiments, providing a powerful tool for research, diagnostics, and personalized medicine. Bioinformatics platforms are the backbone of systems biology research. These software tools manage, analyze, and visualize biological data, supporting everything from sequence alignment to network analysis. As datasets grow, bioinformatics platforms are evolving to handle more data types and provide user-friendly interfaces. Their integration with cloud computing and AI is making advanced analysis accessible to more researchers. Automation is streamlining experimental workflows in systems biology. Robotic systems can perform high-throughput experiments, collect data, and even analyze results with minimal human intervention. This increases efficiency, reduces errors, and frees up scientists to focus on interpretation and innovation. Automated experimentation is especially valuable for large-scale studies and drug discovery. Integrative multiomics combines data from genomics, proteomics, metabolomics, and other omics layers to provide a comprehensive view of biological systems. This holistic approach uncovers relationships that would be missed by studying each layer in isolation. Advanced computational methods are making multiomics integration more powerful, enabling discoveries in disease mechanisms and therapeutic targets. Metabolomics focuses on the small molecules, or metabolites, present in cells and tissues. Recent advances in mass spectrometry and computational analysis have made it possible to profile thousands of metabolites simultaneously. This provides insights into cellular metabolism, disease states, and responses to treatment. Metabolomics is increasingly integrated with other omics data for a systems-level understanding. Proteomics is the large-scale study of proteins, the workhorses of the cell. New technologies like tandem mass tags and data-independent acquisition are increasing the depth and accuracy of protein analysis. Proteomics data are essential for understanding cellular functions and disease mechanisms. When combined with genomics and metabolomics, proteomics provides a more complete picture of biological systems. Transcriptomics studies the RNA molecules in a cell, revealing which genes are active under different conditions. Advances in RNA sequencing have made it possible to analyze gene expression at single-cell resolution and in real time. Transcriptomics is crucial for understanding development, disease, and cellular responses to the environment. Its integration with other omics data is driving new discoveries in systems biology. Epigenomics explores chemical modifications to DNA and histones that regulate gene activity without changing the genetic code. Technologies like bisulfite sequencing and ChIP-seq are revealing how epigenetic changes influence development, disease, and environmental responses. Epigenomics adds another layer of complexity to systems biology, helping explain how identical genomes can produce diverse outcomes. Microbiome, the collection of microbes living in and on our bodies, plays a vital role in health and disease. New sequencing and computational tools are enabling detailed analysis of microbial communities. Systems biology approaches integrate microbiome data with host genomics and metabolomics, revealing complex interactions that influence immunity, metabolism, and disease risk. High-throughput screening technologies allow researchers to test thousands of compounds or genetic perturbations in parallel. Automated platforms and advanced data analysis are making it possible to rapidly identify drug candidates, gene functions, and biological pathways. High-throughput approaches are essential for large-scale systems biology studies and accelerating the pace of discovery. Organoids are miniaturized, simplified versions of organs grown in the lab from stem cells. Tissue engineering combines cells, scaffolds, and growth factors to create functional tissues. These technologies provide powerful models for studying development, disease, and drug responses in a controlled environment. Organoids and engineered tissues are bridging the gap between cell culture and whole organism studies in systems biology. Systems biology is transforming agriculture through precision techniques. By integrating genomic, environmental, and phenotypic data, scientists can optimize crop yields, improve resistance to disease, and reduce environmental impact. Technologies like gene editing, sensor networks, and data analytics are enabling smarter, more sustainable farming practices. Precision agriculture is a prime example of systems biology's impact beyond medicine. Environmental systems biology applies systems-level approaches to study ecosystems and their responses to change. By integrating data from genomics, chemistry, and environmental sensors, scientists can model how organisms interact with each other and their environment. This field is crucial for understanding biodiversity, climate change, and ecosystem health, guiding conservation and sustainability efforts. Systems pharmacology integrates systems biology with pharmacology to understand how drugs affect biological networks. By modeling drug actions at multiple levels, researchers can predict efficacy, side effects, and interactions. This approach is improving drug development and personalized therapy, making treatments safer and more effective. Artificial intelligence is accelerating drug discovery by analyzing vast datasets to identify promising compounds and predict their effects. Machine learning models can screen millions of molecules, optimize drug design, and even suggest new therapeutic targets. AI-driven drug discovery is reducing costs and timelines, bringing new treatments to patients faster than ever before. Systems biology technologies advance, ethical considerations become increasingly important. Issues like data privacy, genetic modification, and equitable access to new therapies must be addressed. Researchers, policymakers, and the public need to work together to ensure that innovations are used responsibly and for the benefit of all. Ethical frameworks are evolving alongside technology to guide decision-making in this rapidly changing field. Rapid evolution of systems biology demands new approaches to education and training. Interdisciplinary programs are teaching students skills in biology, computer science, engineering, and data analysis. Online courses, workshops, and collaborative projects are making cutting-edge knowledge accessible to a wider audience. Preparing the next generation of scientists is essential for sustaining progress in this dynamic field. Public engagement is vital for the success of systems biology. By communicating discoveries and their implications, scientists can build trust and foster informed discussions about new technologies. Outreach programs, science festivals, and accessible media are helping bridge the gap between researchers and the public. Engaged communities are more likely to support and benefit from scientific advances. As new technologies emerge, regulatory frameworks must adapt to ensure safety and efficacy. Systems biology innovations often challenge existing regulations, requiring updates and new guidelines. Collaboration between scientists, regulators, and industry is essential to create policies that support innovation while protecting public health and the environment. Systems biology thrives on collaboration between experts in biology, computer science, engineering, mathematics, and more. Interdisciplinary teams bring diverse perspectives and skills, driving innovation and solving complex problems. Collaborative research networks and consortia are becoming the norm, enabling large-scale projects that would be impossible for individual labs. Future of systems biology is bright, with emerging technologies poised to unlock new frontiers. Advances in AI, multiomics integration, and real-time monitoring will deepen our understanding of life's complexity. As tools become more accessible and data more abundant, systems biology will continue to drive breakthroughs in health, agriculture, and environmental science. Despite rapid progress, systems biology faces significant challenges. Integrating diverse data types, ensuring data quality, and interpreting complex results require ongoing innovation. Ethical, regulatory, and educational hurdles must also be addressed. By working together and embracing new technologies, the scientific community can overcome these obstacles and realize the full potential of systems biology. Cancer research has greatly benefited from systems biology approaches. By integrating genomic, transcriptomic, and proteomic data, scientists can map the complex networks driving tumor growth and resistance. Systems-level models are guiding the development of targeted therapies and personalized treatment strategies, offering hope for more effective cancer care. Systems biology is playing a key role in understanding and combating infectious diseases. By modeling host pathogen interactions and immune responses, researchers can identify vulnerabilities and develop new interventions. The integration of omics data, AI, and high-throughput screening is accelerating vaccine and drug development for diseases like COVID-19. Neuroscience is embracing systems biology to unravel the complexity of the brain. Multiomics, high-content imaging, and network analysis are revealing how genes, proteins, and circuits interact to produce behavior and cognition. These insights are advancing our understanding of brain disorders and guiding the development of new therapies. Plant systems biology integrates genomics, phenomics, and environmental data to improve crop productivity and resilience. By modeling plant growth and stress responses, researchers are developing crops that can thrive in changing climates. These advances are essential for food security and sustainable agriculture in the face of global challenges. Summary: emerging technologies are transforming systems biology into a data-driven predictive science. From omics and AI to wearable sensors and digital twins, these innovations are expanding our ability to understand and manipulate biological systems. As we look to the future, continued collaboration, ethical stewardship, and public engagement will be key to realizing the full promise of systems biology for science and society.